When geneticists at the University of Padua ran DNA sequencing on dust particles vacuumed directly from the Turin Shroud, they cross-referenced their findings against every known global population database on Earth. And not a single living population matched the precise genetic combination recovered from that cloth. That is not a theory advanced by religious advocates or fringe researchers. That is the published finding of a peer-reviewed genetic study. And it is only the starting point of what the science revealed. This video is going to walk you through every layer of that evidence: the DNA, the blood, the fiber analysis, and the institutional record that kept shifting. The Turin Shroud's DNA was finally sequenced. The results matched no living population. When geneticists at the University of Padua ran DNA sequencing on dust particles vacuumed directly from the Turin Shroud, they cross-referenced their findings against every known global population database on Earth. And not a single living population matched the precise genetic combination recovered from that cloth. That is not a theory advanced by religious advocates or fringe researchers. That is the published finding of a peer-reviewed genetic study, and it is only the starting point of what the science revealed. This video is going to walk you through every layer of that evidence: the DNA, the blood, the fiber analysis, and the institutional record that kept shifting every time someone thought the question was settled. The Turin Shroud is a 14-foot linen cloth bearing the faint, full-body impression of a crucified man, frontal image and dorsal image both, laid out head-to-head as if the fabric had been folded over a body lying flat. Millions of people believe it is the burial cloth of Jesus of Nazareth. The Catholic Church has never officially endorsed that claim, and it has never formally denied it either. For 4 decades, the scientific debate has moved back and forth between one camp certain it is a medieval forgery, and another equally certain it predates medieval Europe by more than 1,000 years. The DNA results did not settle that debate. They made it significantly harder to walk away from. The man who led the genetic analysis is Professor Gianni Barcaccia, a full professor of agricultural genetics and genomics at the University of Padua's Department of Agronomy, Food, Natural Resources, Animals, and Environment, one of the oldest agricultural science departments in continental Europe. Barcaccia earned his Master of Science in Plant Genetics and Breeding in 1991 and completed his PhD in the Genetics of Plant Reproductive Systems in 1995, both at the University of Perugia. He has held a tenured professorship at Padua since 2001 and has served as head of his department, overseeing faculty, research funding, and doctoral supervision across multiple genomic programs. He also holds an adjunct professorship in plant breeding at the University of Georgia in Athens and runs Breedomics, a specialist genomic identification laboratory that serves industries where DNA precision is not negotiable: food authentication, seed variety certification, and genetic traceability at a commercial scale. Barcaccia is not a theologian searching for a miracle. He is a working geneticist with more than 2 decades of peer-reviewed molecular analysis behind him, and his team at Padua use the same sequencing tools they deploy routinely on agricultural DNA to examine one of the most analyzed physical objects in human history. Gianni Barcaccia, one Catholic STURP. The genetic material his team worked with was not freshly collected. It came from dust particles vacuumed from the shroud's surface during the landmark 1978 examination carried out by the Shroud of Turin Research Project. Particles that had been preserved in storage for more than 3 decades before Barcaccia's team ever touched them. To understand what the DNA revealed, you first need to understand what the official scientific record says about the object itself. The Shroud of Turin entered documented history around 1350 in Liry, France, when a knight named Geoffrey de Charny put it on public display in a church he had personally built. The local bishop wrote to Pope Clement VII almost immediately, claiming the cloth was a painted forgery. That dispute never fully resolved. The shroud, passed through the hands of the Savoy royal family in the 15th century, survived a fire in Chambéry in 1532, during which molten silver from the reliquary dripped onto the corner of the cloth, was repaired by Poor Clare nuns, who patched the damaged areas with new fabric, and arrived in Turin in 1578, where it has remained under church custodianship ever since. That is the documented provenance. Medieval France, the Savoy court, a fire, patches, then Turin, a clean, traceable European lineage beginning in the 14th century. In 1988, the Vatican granted 3 independent laboratories at Oxford University, the University of Zurich, and the University of Arizona in Tucson permission to perform carbon-14 dating on small cut samples of the cloth. All 3 laboratories returned results pointing to the same window. The linen had been woven sometime between 1260 and 1390 CE. The results were published in the journal Nature. The mainstream press declared the case closed. The Shroud of Turin, radiocarbon science had determined, was a medieval creation, puzzling perhaps, but not ancient, not 1st century, not from Jerusalem. That is the official version. Here is what happened when other scientists examined the same data more carefully. In 2002, textile expert Flury-Lemberg studied the shroud's seam construction and identified a stitching technique so specific that the only comparable example she could locate was found on a 1st century textile excavated at the Jewish fortress of Masada in Israel. Then, in 2005, chemist Raymond Rogers, a member of the original 1978 examination team, published a study in the peer-reviewed journal Thermochimica Acta, arguing that the samples cut for the 1988 carbon dating had come from a corner of the cloth that had been repaired during the medieval period, a section reinforced with newer cotton threads that were invisible to the naked eye. The chemical tests Rogers applied to fibers from the main body of the shroud, not the repaired corner, returned an age range consistent with a 1st century origin. In 2022, Dr. Liberato de Caro of Italy's Institute of Crystallography at the National Research Council in Bari published results from wide-angle X-ray scattering analysis, a method that measures structural degradation in cellulose fibers as they age. De Caro's team compared shroud fibers against samples from known dated linens, including a Masada fabric verified to originate between 55 and 74 CE. The shroud's fiber degradation pattern matched closest to that 1st century Masada sample, not the 1260 to 1390 window the carbon dating had produced. The analysis indicated a cloth approximately 2,000 years old, stored in conditions of roughly 22 degrees Celsius and 55% relative humidity for the majority of its existence. Bioarchaeology, none of these studies proved. The shroud was the burial cloth of Jesus. What they collectively did was establish that the 1988 carbon dating result, the result that was supposed to end the argument, was being challenged by multiple independent lines of evidence using entirely different scientific methods. Then came the DNA. The 1978 STURP examination was the most extensive hands-on scientific investigation ever conducted on the shroud. More than 30 scientists, engineers, and medical professionals from the United States and Europe were granted direct access to the cloth for 120 consecutive hours inside the royal palace adjoining Turin Cathedral. They worked around the clock, running X-ray fluorescence, ultraviolet and infrared photography, spectroscopy and microscopy simultaneously on a cloth stretched flat on long examination tables under laboratory lighting. They took adhesive tape samples from dozens of surface points across the fabric and documented every contact point and procedure. Those samples were preserved in controlled conditions. It is from those 1978 samples that Barcaccia's team extracted the genetic material for their 2015 analysis. Shroud Zoan. What the STURP team had already established before the DNA work began was deeply difficult to explain. The blood on the shroud, present in substantial quantities corresponding to wounds on the head, both wrists, the feet, one side of the torso, and the back, had been tested and found to belong to blood type AB. That was notable. What was harder still to account for was the image itself. After 120 hours of testing, the STURP team's formal 1981 conclusion stated that the body image on the shroud is not painted, not dyed, not printed, and not produced by any applied substance. The image is formed in the topmost fibers of the linen threads, penetrating no deeper than 2 or 3 fiber cells, and it encodes 3-dimensional spatial information about the body it covered. Information that no photographic or artistic process available in medieval Europe could have deliberately produced. The STURP team was not assembled from believers. The group included physicists, chemists, imaging specialists, and forensic pathologists. Several arrived as open skeptics. Barry Schwartz, the official STURP documentation photographer, was a Jewish man with no prior investment in the shroud's authenticity, who has described spending years after 1978 searching for a natural explanation for what his team documented, and failing to find one convincing enough to publish. YouTube. Against that backdrop, Barcaccia's team began extracting and sequencing the DNA. The first question they were trying to answer was geographic. Where had the cloth physically been? Every hand that touched the shroud, every person who pressed a face against it during centuries of public display, every individual who handled it in storage or transport, all of them potentially deposited microscopic genetic traces on the linen surface. By identifying which haplogroups the DNA sequences belonged to, the researchers could theoretically reconstruct the cloth's movement across populations in time. The 2015 study identified 93 distinct mitochondrial DNA sequences corresponding to material left by numerous individuals. Mitochondrial DNA passes from mother to child and clusters into haplogroups, genetic lineages that track to specific geographic populations across generations. The haplogroups recovered from the shroud ranged across an enormous geographic spread: Western Europe, the Near East, North Africa, East Africa, and East Asia, with sequences associated with populations in India and China. Given centuries of public display and handling by pilgrims from across the known world, some degree of genetic diversity on the surface was expected. The plant DNA was not pollen, and plant genetic material extracted from the shroud traces included species indigenous to the Middle East, the Eastern Mediterranean, and North Africa, confirming findings from pollen analysis conducted by Swiss criminologist Max Frey-Sulzer in the 1970s, who had identified dozens of plant species on the cloth, some of which grow exclusively in the area around Jerusalem. Barcaccia's molecular analysis confirmed Frey's results from a genetic level, adding specific detail about the plant lineages and deepening the geographic picture considerably. PMC.NCBI.NLM.NIH-1. The human haplogroup data pointed toward a cloth that had traveled through the Mediterranean basin and the Middle East before reaching Europe, a pattern consistent with a documented origin in the ancient Near East. But the combination of haplogroups present, taken as a collective genetic fingerprint, did not correspond to any single modern population. The researchers cross-referenced every available global population database and found parallels to individual lineages in living groups. But the specific arrangement of haplogroups, in those proportions, in that combination, had no exact match anywhere in the record. That was the finding from the first version of the study. The second version, from the 2026 metagenomic reanalysis using significantly more powerful sequencing technology, produced a result that no one had anticipated going in. Barcaccia and his collaborators returned to those same 1978 dust samples with next-generation sequencing tools unavailable to them a decade earlier, capable of identifying and reconstructing fragmented DNA at resolutions the 2015 study could not approach. The new analysis found that approximately 40% of the identifiable human DNA on the shroud traces to Indian lineages, specifically haplogroups associated with populations on the Indian subcontinent, not Western Eurasia, not the Near East, India. What this means, and what it does not mean, requires precision. This is not proof the shroud originated in India. It does not establish anything directly about who the man on the cloth was or where he lived. What it reveals is that the genetic record left on the linen by every person who handled it across its documented and undocumented history carries a 40% Indian genetic signature that has no straightforward explanation if the cloth's documented history is the complete history. If the shroud appeared in France in 1350 and had only passed through European hands before that, you would expect a predominantly European genetic profile. A dominant Indian signature is not what the documented chain of custody predicts. The written record we have does not account for it. The 2026 study also identified radiocarbon dating from specific textile residues associated with the shroud's reliquary, residues tied to known repair and storage periods that return dates between 1451 and 1800 CE. That finding applied to repair material, not to the main body of the cloth. That distinction tends to evaporate in the published headlines. If you have never followed this particular body of evidence before and are finding it more difficult to dismiss than you expected, subscribe now, because documented, evidence-based cases where the primary record and the official summary point in different directions are exactly what this channel covers every week. There's one more thread that rarely surfaces in the main discussion. 3 separate objects, each claimed by different parts of the Catholic world to have been in direct physical contact with Jesus of Nazareth: the Shroud of Turin, the Sudarium of Oviedo, and the Tunic of Argenteuil, have all been independently tested for blood type. All three returned the same result, AB, the Sudarium of Oviedo, a cloth kept in a cathedral in northern Spain and listed in a church inventory as early as the 7th century, bears over 120 forensic correspondence points with the bloodstains on the Turin Shroud, matching wound locations, matching blood transfer patterns, and a matching impression of a beard and hair arrangement consistent with the image on the Turin cloth. The probability of that degree of forensic correspondence arising by coincidence between two separately produced medieval forgeries, according to analysts who have studied both objects, is statistically negligible. None of this constitutes certainty. The scientific literature on the shroud represents a genuine ongoing dispute, not a settled case dressed in the clothing of an open question. There are serious researchers who maintain the 1988 carbon dating is accurate, and that every alternative explanation is selective reasoning applied after the fact. There are equally serious researchers who argue the dating was performed on a repair patch, and that every other analytical method, fiber degradation, pollen analysis, seam construction, forensic blood comparison, points to a first-century origin. The image formation mechanism remains formally and officially unexplained after 47 years and hundreds of thousands of laboratory hours invested by some of the most qualified analysts in the world. What Barcaccia's DNA work added to all of this is not a definitive answer. It added a specific, measurable anomaly that sits inside the data and will not resolve itself with the available evidence. A genetic fingerprint that crosses continents in a way the cloth's documented history cannot account for. A 40% Indian genetic signature on a relic supposedly restricted to European custody. A haplogroup combination that does not match any living population on record. The cloth is currently held in a temperature-controlled, climate-stabilized vault beneath the Cathedral of St. John the Baptist in Turin, made available for public display on rare occasions. The last major exhibition drew over 2 million visitors across several weeks. The next direct examination of its fibers requires Vatican authorization. Further DNA work has been discussed in the academic community, specifically the possibility of extracting nuclear DNA rather than only mitochondrial DNA, which would provide far more precise individual identification and geographic tracing. That authorization has not been granted. Every scientific tool applied to this cloth has returned results that were supposed to simplify the problem and instead deepened it. The carbon dating was supposed to close the argument. The X-ray analysis pulled it back open. The DNA sequencing was expected to confirm a medieval European origin. It produced a genetic profile with a 40% Indian signature and no match in any modern population database on Earth. The image on the cloth shows wounds on both wrists, not the palms as centuries of Western religious art depicted crucifixion, but the wrists, which is where forensic and historical analysis confirms a nail would actually need to be placed to bear the weight of a body. That anatomical detail was not part of common knowledge in medieval Europe. The wound positioning, the directionality of the blood flows, the presence of both frontal and dorsal images encoded with three-dimensional spatial information. The STURP team examined all of it across 120 hours of continuous testing in 1978 and still could not identify the mechanism that produced the image. The question the DNA findings leave sitting in the room, the one no database has resolved and no paper has answered, is this: if you sequence the genetic material on the most examined, most disputed piece of cloth in recorded history, and the combination of haplogroups you recover does not match any living population on Earth, what does that tell you about where the cloth actually came from? That is the question for the comments. Whether you read this as a genuine relic, a medieval production of unknown method, or a scientific problem that keeps generating data without generating resolution, the DNA does not care what conclusion you arrived with. Leave your reading below. This argument has been running for 6 centuries, and the evidence keeps finding new ways to make it harder to close. If you want to keep going on stories like this one, documented cases where the physical evidence and the official record do not line up, subscribe and stay with us. There is considerably more where this came from.